Hi, my name is Laura Johnson, and I'm here to present on the master's thesis research that I completed at the University of Oregon's Environmental Studies program, as advised by Doctors Lauren Hallett and Patricia McDowell. And I wanted to understand how land use and stream hydraulics interacted to structure freshwater mussel distribution, abundance, and demography in a Pacific Northwest watershed. And what I've learned by recording this multiple times now is that I have way too much to say and not enough time. And so I'm going to feel like I am skimping on the details in order to fit this into 12 minutes. And please feel free to ask more questions and get some follow-up from me in the chat. So in my research, I wanted to confront the reality that we have a modern world in which natural and anthropogenic influences are structuring our mussel abundances and distributions. And this is happening at the patch scale of centimeters to meters, the reach scale of tens to hundreds of meters, and the watershed scale of 1 kilometer and greater. And a natural control of mussel abundance and distribution is hydraulics. And in the Pacific Northwest, high energy gravel stream beds in particular, hydraulics are likely influencing mussel abundances and distributions as a first order control at both the patch and the reach scale. Now, at all scales, land use is important, and land use has been identified, other than dams, as one of the most pervasive threats to mussels. And this is largely due to the way that it interacts with a myriad of other processes in altering flow regimes, increasing influx of sediments and pollutants into streams, degrading riparian zones, just to name a few. And so land use is this really complex thing that triggers all these other processes that interact with mussel habitat at multiple scales. These two lenses are what I viewed my research project through, and I created a couple hypotheses. First, at that reach scale of tens to hundreds of meters, I hypothesized that as stream power increased, mussel distribution and abundance would decrease, and that mussel abundances would be decreasing with increasing stream power. And I chose to investigate stream power as my hydraulic variable, which stream power is the rate of potential energy expenditure against channel bed and banks for a specified length of channel. And it's highly related to sediment transport processes and had the benefit of being able to be calculated remotely from the office. Shear stress is another variable commonly used, but it involves a lot more intensive field measurements. And I really wanted to see what applicability and relationship stream power might have in this context. My second hypothesis was that land use is structuring mussel abundance and population structure at the watershed scale. And I expected to see lower mussel abundances, and in particular, a lower percentage of juveniles in bed populations as anthropogenic land use increased. And at the watershed scale, I delineated a drainage basin area for each specific mussel site. So this is an example of one mussel site that would be located at the intersection of the light and the shaded region at the stream, the South Umpqua stream low point. And USGS stream stats was used to delineate each drainage basin area, and it's all of those upstream, upland areas contributing to a point in the stream. Now, my third hypothesis was that anthropogenic land uses would supersede stream power at some threshold level of change, and that mussel abundances would differ in response to stream power only in reaches in which land use impacts were not already a limiting factor. I investigated these questions and hypotheses in the South Umpqua River Basin, Oregon, which is located primarily in Douglas County in southwestern Oregon. This basin has a Mediterranean climate with dry summers and wet, cold winters. There are no dams on the South Umpqua River, which is a big benefit, which the host fish species that mussels need are widespread. This is a really gravel-rich geomorphic province that the South Umpqua River drains. So there's a lot of gravel habitat around for mussels to use. I further delineated this basin into 3 zones: the lower South Umpqua River, which is downstream from Canyonville, where the majority of the population and anthropogenic land use is; Upper South Umpqua River refers to upstream from Canyonville to approximately Tiller. And the third zone is Cow Creek, which is the largest tributary of the South Umpqua River. I selected 11 sites on the South Umpqua River and 2 on Cow Creek, a priori from a recent database records that indicated mussel presence at a site. And I did a series of things to get my data. The first was to complete a site assessment where I used snorkel survey methodologies to survey all available habitat, spending at least 5 person-hours per site. I defined a mussel aggregation as one or more mussels in close proximity to each other. Different aggregations happened if there were more than 10 meters of unoccupied substrate, or aggregations were in substantially different habitat zones. And a mussel bed was 15 or more mussels visible at the surface within an aggregation. I marked a GPS point for all of these aggregations and beds, and then I followed up the bed locations with systematic sampling. I surveyed 3 Margaritifera falcata beds, and the rest of my analyses are going to focus on Margaritifera falcata. The bed that I surveyed in the lower South Umpqua River actually didn't meet my definition of bed set by the project. There were only 11 animals in the aggregation, but this was the highest abundance aggregation in the entire South Umpqua River. So I went ahead and sampled it like it was a bed. There was an Onodonta bed that I sampled in the upper South Umpqua River at the same site as the Margaritifera bed was sampled, but because of time constraints, I'm not going to discuss that one further here, and would love to follow up with folks if you wanted to know more about that. I use quadrats as my sampling unit. I set them along transects. I either censused a bed entirely or systematically sampled it based on bed size and initial density estimates. I did the adequate number of excavation, assuming 50 to 60% of mussels would be available at the surface. I excavated to 13 centimeters depth and sieved through 3.5 millimeter mesh screens. We measured all animals along their longest axis with calipers and identified the species or clade level. My project used length as a proxy for age to avoid destructively sampling animals. Because the correlation between length and age is approximately linear for young mussels, we thought this was an appropriate method to answer questions about reproduction and juvenile size classes. 3 centimeters was the upper size limit for a juvenile Margaritifera that we found based on laboratory growth experiments. At each site, we developed a generalized specific stream power value, which is watts per meter squared, using remotely collected LiDAR data and ArcGIS, and the 10-year peak flow discharge at each site. And we recognize that this is just a generalized value to represent hydraulic disturbance at each reach, and isn't actually supposed to indicate the actual stream power value being felt in any particular place in the stream. So it's really not a patch level, but a reach level value. And then our land use categories were derived from the USGS's National Land Cover Database. From 2016 imagery, we reclassified that into 7 categories, which are seen here on this map. And we focused on forest at the drainage basin scale as our variable to put into our modeling, because all of the other anthropogenic land uses were highly correlated, and that as forest cover decreased, all of those other anthropogenic uses increased. Research facts of note: we searched 4.2 kilometers of the lower South Umpqua River, 2.53 kilometers of the upper South Umpqua River, and 1.38 kilometers of Cow Creek. We found 49 mussel aggregations, including 15 beds across 13 sites. All 3 genera of Western freshwater mussels were found: Margaritifera, Ganidia, and Onodonta. And we found really high densities of Asian clams, Corbicula fluminea, in the lower South Umpqua River. And in the lower South Umpqua River, where those really high densities of Asian clams were, there was also the lowest mussel abundances. And so in the little picture at the top right, the red circle is the lower basin, and that corresponds to the red dots on this graph. And then the blue zone is the upper South Umpqua River, and that corresponds to the blue dots. So as you moved upstream, you got much higher abundances, and you lost those Asian clams at a certain clinch point in the river. And this is just a picture of what one of our excavation units looked like on the lower South Umpqua River bed that we sampled. And that land use category that we used of forest, as forest increased in that drainage basin area, the mussel abundances increased. And then as anthropogenic land use increased in the lower river, mussel abundances decreased. And at that reach scale, stream power value failed to explain variation in our data. It ended up being a significant factor in our model, but didn't actually have very much ecological significance in terms of its values. And either that is indicative that this isn't a very good explanatory variable and wouldn't have good predictive power, or something that I thought about was that some of our highest stream power sites that I highlighted here in red, when we were surveying in the field based on our observations, these sites appear to be dominated by really patch scale variables, where the high densities of mussels were only located behind flow refugia. And so potentially this reach scale stream power value is just really not able to predict abundance that is dictated by these smaller patch scale variables, which was something interesting to think about. There was successful Margaritifera recruitment throughout the basin. This blue thump is our bed in the upper South Umpqua River. The red is the lower South Umpqua River, and green is Cow Creek. And juveniles were present everywhere, but higher populations were in the upper South Umpqua River. And in our excavation units, overwhelmingly, we were pulling younger mussels out of excavated beds. So it was important to excavate. For the future, we would want to systematically survey additional Western pearl shell and floater mussel beds, do repeat monitoring, increase the number of site assessments in all portions of the river, investigate hydraulic variables at multiple scales, understand the impact of Asian clams on native mussels, and establish age-length relationships for the basin. Thank you for your time.